appreciate you being here, and I am going to be presenting my undergraduate research on morphological variation in smallmouth buffalo. This project was completed in partnership with Francis Marion University, Wright State University, as well as the North Carolina Wildlife Resources Commission. Buffalo fishes are native to the Mississippi River Basin and are among the longest lived fishes in North America, some individuals reaching over 100 years of age. There are three species of buffalo. There's the smallmouth buffalo, the bigmouth buffalo, and the black buffalo. They are benthic feeders that consume vertebrates, algae, and insects, and play a primary role in transferring nutrients as they migrate to spawn. In addition to their ecological importance, they provide a commercial fishery within their native range. However, this fishery has seen overexploitation and has been affected by habitat loss due to deforestation. In response, fisheries managers are attempting to restore and conserve buffalo species. This is particularly the case in western North Carolina, where managers are attempting to rebuild the smallmouth buffalo population in response to habitat loss due to deforestation. They're completing this by translocation. However, possible hybridization with black buffalo within that system has made it difficult for them to identify individuals that maintain the original smallmouth buffalo form. In contrast, the smallmouth buffalo was introduced to South Carolina in the Great Petey River in 1919. Here, since black buffalo are absent from the system, the smallmouth buffalo have been isolated from the effects of hybridization for a little over 100 years. As a result, the population of smallmouth here are considered to have the original smallmouth buffalo form. The French Broad River and the Great Petey River are the two river systems we explored in the study. And as I move forward, I'll be referring interchangeably to the French Broad River as North Carolina and the Great Petey River as South Carolina. So to understand the importance of our work, we must first look at why morphology matters. Morphology gives us a window into population differences across systems. It helps us to determine whether our field identification is reliable, especially in systems where hybridization is suspected. It also allows us to distinguish between species and hybrids. And finally, it reflects the evolutionary and the ecomorphological pressures that affect things such as swimming performance, habitat use, and even feeding strategies. So to explore morphology in this study, we employed a geometric morphometric analysis, and we set out two clear objectives. First, we sought to characterize the overall body shape of smallmouth buffalo, both in North Carolina as well as in South Carolina. And second, we sought to compare these regional morphological patterns to determine if these two populations differ in consistent and biologically meaningful ways. We began our study by collecting 101 smallmouth buffalo, 44 from North Carolina and 57 from South Carolina. All individuals were collected in the spring of 2024 using electrofishing as our means of capture. A geometric morphometric analysis was applied to each individual, followed by a series of statistical analyses to test for shape variation. We made trait-based characteristics as our basis for identification in the field. These listed characteristics were derived from a publication by the University of North Carolina regarding their freshwater fishes in North Carolina. As you can see, this field-based identification can prove challenging as the smallmouth buffalo and the black buffalo share overlapping characteristics. However, the most distinguishing feature between the two is the overall body depth of their shape. The smallmouth buffalo has an expanded deep body form, whereas the black buffalo has a more compressed, elongated form. Once individuals were identified as smallmouth buffalo in the field, we took lateral photographs of each. We placed 35 anatomical landmarks on each picture. Of these, 19 were sliders and 16 were fixed landmarks. This process was completed using the TPS suite of software. Then we applied a generalized Procrustes analysis, which standardizes the size, position, and also orientation so that only differences in shape remain. We analyzed the landmark data in R using the GeoMorph package. We then summarized the major axes of variation using a principal component analysis. We then interpreted these axes using a scree plot as well as a percent variation explainer. We then applied a broken stick model to determine which of these axes were significant and And finally, we tested the effects of total length and location on shape using Krusty's linear regression. Here we see a total length distribution by location, and as it indicates, North Carolina saw a minimum length of 428 millimeters and a maximum of 698. South Carolina had a minimum of 344 millimeters and a maximum of 935 millimeters. Now, while South Carolina saw a larger range of total length distribution, most measurements for both locations fell within a similar size window. Here are the results of our principal component analysis. On the left, we have a scree plot that lists the principal components that best describe the overall variation within our data. It indicates that the first principal component explains the majority of our variation. On the right, we have a scatter plot where each point represents an individual fish. This scatter plot separates individuals based on where their body shape falls along these axes of variation. Together, principal components 1 and 2 account for 57.58% of total variation. However, as our scree plot indicates, the majority of this variation comes from PC1, which is 41.51 total variation. So what does this variation look like along these axes? Well, these deformation grids give us a two-dimensional representation of what body shape looks like at the minimum and maximum end of each axis. Since we established PC2 accounts for little variation and as a result subtle body shape differences, we will spend our time looking closely at PC1. On the left, we see a deformation grid that gives us a body shape representation at the minimum end of PC1. Here, this body shape is more compressed and elongated when compared to the maximum end, which gives us a body shape that's more expanded and deep. So this means that the further left on our scatter plot a fish is located, the more they will exhibit a more compressed and elongated shape, and the further right they are on our scatter plot, the more deep and expanded their overall body shape will be. So when we overlay location over individual fish on our scatter plot, a pattern emerges. North Carolina and South Carolina smallmouth buffalo form distinct clusters, and since we established elongated, compressed forms are on the left and deep, expanded forms are on the right, we can see that overall body shape separates into regional groupings, with North Carolina on the left and South Carolina on the right. This is an indication that these two populations are morphologically distinct from one another. So even though we can see this regional divergence, we needed to test these differences in order to determine their statistical significance. This is where we applied our Krusty's linear regression to answer two important questions. First, is fish shape related to total length? With a p-value less than 0.01, the answer to that was yes. And that's not surprising, as allometric growth is consistent with most fish growth patterns. Simply put, fish tend to change shape as they grow older and longer. However, the second question is the important one we're looking at, is after accounting for the differences due to total length, is shape related to location? And again, with a p-value less than 0.01, the answer to that is yes. Location has a statistically significant effect on body shape. So what does statistical significance mean in real life application? Well, as we have here on the left, we have an individual from our population in North Carolina. On the right, we have an individual from our population in South Carolina. When we compare side by side, we can see that our statistical significance translates directly into biological significance. To summarize our major findings, most variation within our data was captured by the first principal component axis. In North Carolina and South Carolina, smallmouth buffalo form distinct clusters along this primary axis. And most importantly, North Carolina smallmouth buffalo tended to be more compressed and elongated, while South Carolina tended to be more expanded and deep-bodied in form. Now, it's true that other factors can contribute to this regional divergence, such as flow regime, as well as resource partitioning. However, current literature suggests that hybridization in the French Broad River is most likely the driver to this regional divergence. So I believe our findings have two major implications in fisheries management, especially for smallmouth buffalo. Possible hybridization makes species level identification difficult. This means that in the field, managers may not be able to rely solely on morphology to make determinations between smallmouth and black buffalo. This uncertainty can lead to problems with population assessments and monitoring accuracy, or any decisions tied strictly to species identity. And secondly, the regional divergence that we witnessed suggests that these two populations are not interchangeable. Morphological, as well as more than likely genetic structure, differ between the two systems. So when making decisions based on restoration and supplementation, local population characteristics should be considered rather than assuming uniformity across species range. Looking ahead, I believe there's two areas that we can expand our research. First, we can do a species-to-species comparison by expanding our geometric morphometric analysis to include both smallmouth and black buffalo. We can maybe determine the shape variables that best distinguish the two species. And we can also broaden our genetic work by incorporating additional markers or targeted primers. Maybe we can resolve some regional differences and thereby detect species boundaries as well as highlights. With that, again, I would like to thank my research partners, my field collection crew, and our funding contributors. Most of all, thank you all for your time and your attention. I'm glad to take any questions.